So, uh, so this is for the specifically for the focused questions, for the focused informal practice questions. For the first KPI 3.1.2, we are going to be speaking about the roles of the DNA tools. Specifically, we're going to be speaking about restriction enzymes, sticky and blunt ends, plus gel electrophoresis. So for this drawing, he's asking you to use this diagram here to answer questions 1 to 3. For question number 1, which enzyme is used in step number 2? So if you look here, in step number 1, he is extracting the DNA from either the animal cell or the plasmid from the bacteria. And in step number 2, he is splicing the DNA. He is cutting this DNA down into smaller pieces. So what do we use to cut the DNA? The answer is very easy. What we use is a restriction enzyme, because the restriction enzyme is the enzyme that is used to cut the DNA down. In question number 2, which enzyme is used in step number 3? Now, what do we use? Which enzyme do we use to attach the human gene to the bacterial plasmid? We use the enzyme called the DNA ligase. Okay. And question number 3, which type of ends are produced in step number 2? What we have produced here are sticky ends. Please remember this. Sticky ends are if we have a DNA like this, okay? If we cut the DNA with one longer piece and one shorter piece, like this, these are called sticky ends, okay? If we cut it like this, sharp like this, it will be called blunt ends. Bas. Now, for the next KPI, here, as you can see here in front of you, we have a gel electrophoresis. Okay, so use the diagram below that shows how DNA is separated to answer questions 4 to 7. Question number 4. Which of the following techniques is used to separate the DNA fragments? We are using, we are using a gel plus electricity. So the technique is called the gel electrophoresis. Okay. Number 5. Which of the following is correct about the two fragments labeled as X and Y? أول شيء X, the band X, is it negative or positive? I am sure it's negative charged. DNA is always negatively charged. So C is wrong لأنه it's positive, و D is wrong لأنه it says it's positive. It's wrong. But X is negative or X is negative. Here is correct. And the X has moved in a distance shorter than the Y has moved. So the X is bigger. Okay? So X is negatively charged and bigger than Y in size. Yes, that's correct. But it is smaller غلط. So A is the correct answer. Okay? Number 6. Which two children are most likely to be biological siblings and are children of the father and mother shown in the diagram? Let's see. Here is the father. Here is the mother. Which two are the children here? For example, here this is child 1. It has one matching with the father, with the other one matching with the mother. So it is a child of the father and the mother. It is a probable child. Child number 2. It has one piece matching with the mother, with the other piece matching with the father. So it is a child as well. Child 3. This piece, this DNA fragment here is not matching with the father, not matching. لا الfather ولا المother. With this piece over here not matching the father or the mother. So here it cannot be the child of the mother and the father. With child number 4, the same. This piece here is not matching the father or the mother. With this piece over here is not matching the father or the mother. So number 3 and number 4 cannot be children of the mother and the father. Only 1 and 2. But all you have to do is just look and see them. I will show it to you again. 1 second. I will erase everything, highlight. We'll use the highlighter. So look here. Here is the child number 1. This fragment here is matching with this fragment here. And this fragment here is matching with this fragment here. So they are one with the father, one with the mother. For child number 2, this fragment here is matching the X for the mother. This fragment here is matching this fragment for the father. So they are the children. These here are not matching anything in the mother or the father. So they cannot be the children. Okay? Number 7. Name 2 factors that are used to separate the DNA fragments. There are different factors. For example, you can simply say size, you can say charge, you can say different things over here. Okay? Now, KPI number 4. Sequence using diagram and models how recombinant DNA is made and manipulated. You have to include the role of the plasmid and DNA ligase. Question number 8. Which of the following is the correct sequence of steps of the process of recombinant DNA? Yani, what are the correct steps that we use to make recombinant DNA? So if we start with DNA cleavage, that yani DNA splicing, DNA cutting, this is step number 1. That's correct. Then this is 1. Then DNA joining by DNA ligase, this is step number 2. Then introducing the DNA into the host cell to a plasma. طبعا, this is step number 3. So A is the correct answer. And here is the correct sequence. Pay attention to this. All the others are incorrect. Then, the diagram below shows the steps of producing a recombinant DNA. Use the diagram below to answer questions 9 and 10, please. So it is the same picture as the one that we have seen before in the multiple choice. You might get it as a multiple choice, or you might get it as a free response. Question number 9, let's see. The diagram shown as an example of recombinant DNA technology. Define recombinant DNA. You just have to say the definition of recombinant DNA, and that is something that you have to memorize. Recombinant DNA is a DNA, for example, that results from joining 2 segments of DNA from 2 different sources. So here we have DNA from an animal, and we have DNA from bacteria, and we join them together to produce the recombinant DNA here. So this is the definition. Bas. Question number 10. Complete the table below to describe the steps 1 to 4 shown in the diagram. Say all the steps that are shown in the diagram. So what is happening in step number 1, step number 2, step number 3, step number 4, and so on? You have to memorize them. They are always going to be the same. إذا بتستخدموا, for example, if you change here animal cell to human cell to bacterial cell, anything, it will always be the same steps. So you just have to memorize them. Step number 1, animal cell genome and bacterial cell plasmids are isolated. So I will show you here. So here, and in step number 1, I isolated the DNA from the animal cell and isolated the DNA from the bacterial cell. This is step number 1. In step number 2, the cell genome and bacteria are both cut with the same restriction enzyme. So I cut the human DNA with the same restriction enzyme, and I cut the bacterial plasmid with the same restriction enzyme. Step number 3, the desired gene and the plasmid are joined together using ligase enzyme. How do I join them together here in step number 3? Using DNA ligase. This is something we already talked about in this recording. With step number 4, I introduce this plasmid into the, you can say, for example, here he used the word into the host cell. If you could go into the new bacterial cell, all are correct. Okay? Now, question number 11. Use the diagram which summarizes the steps of the process of polymerase chain reaction PCR to answer questions A to C. Let's see. So here he is telling you we have PCR. We have step number 1. You already know it. You should memorize it. Step number 2 and step number 3. أول شيء. Number A. What is the purpose to do this process in biology? You have to say it is used to amplify the sequences of DNA, to amplify the number of the sequences. Okay? So مثلا, or you can simply say it helps to produce millions of the copies of the specific DNA. In our PowerPoints or in our book, we use the word amplify. Here, to produce millions of copies of the DNA. That's very good. B, what is the name of the component binding with the DNA strands in step 2? What's the name of this component here? and this component definitely is a primer. Okay, so you can simply say it's a primer or RNA primase. Okay, and then C, you have to write the steps. You have seen this same question in the quizzes, and we have seen it multiple times in the PowerPoint. So you know them. Step number 1, it is called the denaturation step, and it happens at almost a temperature of 95 degrees Celsius. You should never say 100. Step number 2 is the primer annealing. It happens at this almost a stage of 55 degrees Celsius. You can say a range as well. Pay attention to the range. And the extension, it happens usually at 72 degrees Celsius. And you have to say the description. What happens in each step? Denaturation, both DNA strands separate from each other, as you can see here. They are separated from each other. Step number 2, the primer by the temperature decreases from 95 to 65 degrees Celsius, and the primer binds to each DNA strand. And the last step, the RNA polymerase binds to the DNA and extends the new strands but in both directions, in both opposite direction. At the end, I have started with one DNA, I will end up with 2 DNAs. Okay. No. Now, starting with the second lesson here, define the term genomics. What is the meaning of the word genomics? If you go quickly here, the genomics is simply the study of the organism's genome. Okay. Then identify the goal of the Human Genome Project. What was the goal of the Human Genome Project? Was it to facilitate the development of new therapies? It wasn't directed to make new therapies. Sequencing the entire genome of 3 billion nucleotides. To correct the mutated genes that cause human diseases, to identify all human genes. The correct answer is B. Okay, sequencing the entire genome of the 3 billion nucleotides. This was the only goal for the Human Genome Project. Okay. أو حتى even you can speak about this is correct as well, but this was something that they discovered later on. They used the Human Genome Project, the result of the Human Genome Project, to know all the genes, but the best answer for this question is B. Then specify the application and benefits of gene therapy. What do you use the gene therapy for? So, question 14. Cystic fibrosis is an inherited disorder that causes severe damage to the lungs, digestive system, and other organs in the body. So he's here telling you about a disease called cystic fibrosis that causes many problems in the body. Which of the following tools would be a better option for treating individuals who are affected with cystic fibrosis? Haplotyping, we don't need it. Proteomics, of course not. We are not going to change the protein of the person. Organ transplantation, we cannot, even if we change the organ, we do not change the gene of the individual. So the best thing we can do for these people are a gene therapy, which doesn't exist as of today. Okay? Then identify the aim of gene therapy and one way in which it can be done. This is one example of how it can be done mentioned in this question. We are going to write a different example. For example, gene therapy is a technique aimed at correcting mutated genes. So, but the only answer I care about here is correcting mutated genes, okay, that cause a human disease. How it can be done? Scientists, but how it is done? Insert a normal gene into a chromosome to replace a dysfunctional gene. So, and all I'm doing is removing an abnormal gene, replacing it with a normal gene. This is what is gene therapy. Okay? Now, question number 16. Which of the following are among the conditions that are necessary for natural selection to occur? This is a new lesson. Natural selection. Now, inheritable adaptations vary among individuals. Yes. Number 1, we have variation. Number 2, more individuals are born than can survive. Yes, this is one condition كمان. Reproductive advantage. 3, the environmental conditions are stable. غلط. That's not a correct answer. So Anna, what I want the answer to be is number 1 and number 2 only, which is C. Okay, remember, what are the conditions? Here we have 4 conditions that you have to memorize. Number 1 is variation, number 2 is heritability, number 3 is reproductive advantage, number 4 is, what's number 4? Variation, heritability, reproductive advantage, and variation. Did I say it correct? I said it already. Well, I forgot the 4th one. Check it in the PowerPoint. These are very important. All these 4. Now thinking of the top of my head, I cannot remember. We have them here. We have them here. Describe the principles illustrated in the diagram below. So we have variation, number 1. Number 2, overproduction. Number 3, heritability. Number 4, reproduction. You have to be able to describe them. Okay? For example, here you have number 1, variation. Number 2, overproduction. Number 3, heritability. Number 4, reproductive advantage. Okay? Now, differentiate between evolution and natural selection. What's the differences between them? Simply speaking, evolution is the cumulative changes in a group of organisms over a long period of time. Evolution, different small changes that have grouped, accumulated, piled up in one organism over long period, millions of years. Whereas natural selection is the process whereby organisms better adapted to their environment tend to survive and produce more offsprings. Natural selection is the mechanism by which evolution occurs. Now, I want you to keep in your heads, natural selection, usually it's not, evolution takes a very long time. It's a huge accumulation of different changes that occur in one individual. Natural selection, not as much. It only leads to the survival of the fittest. But keep in your heads, this is very important to say, natural selection is the mechanism by which evolution occurs. Natural selection with natural selection with natural selection in the same individual over millions of years is what leads to evolution. Okay? So that's something that you need to remember. Now, question number 19. Which of the following examples can be used to illustrate artificial selection but not natural selection? Let's see. In a population of moths, so I want to see which one is artificial selection in selective breeding, not natural selection. In a population of moths, darker colored moths become more common because they blend into soot-covered trees, avoiding predators. So darker colored moths avoid predators because they stand on the trees. This is natural selection. So this is not the correct answer. This is not artificial selection. B. Wolves evolve thicker fur over generations to survive colder climate. He's saying because the environment is getting colder, the wolves are developing longer body hair, which is natural selection. So this is not the correct answer. This is natural selection. C. Certain bacteria develop resistance. Resistance is a natural selection. ببساطة. So this is not artificial selection. But which one is artificial selection? This is what he's asking for. Farmers breed cows that produce more milk to increase dairy products in the herd. No, of course, this is artificial selection. And you are choosing to produce cows that produce more milk. So this is definitely natural selection. Now, number 20. Which of the following statements correctly differentiates between derived traits and ancestral traits? Remember, this is a very important KPI for this lesson or for the next lesson. Remember, derived traits are traits that exist in the current generation, whereas ancestral traits are traits that exist in the ancestors only. Okay? So, number 20. Derived traits are inherited from common ancestors, while ancestral traits appear only in modern species. بالعكس. So this is the opposite. So A is wrong. B, ancestral traits are found in both the common ancestor and its descendants, while derived traits are unique to certain descendants. That sounds good. Let's read the others. Derived traits are shared by all species in a lineage. غلط. Not by all species, only by the new species. Ancestral traits develop after a species diverge from its ancestor. No, because it should be with the ancestor. So the only correct answer in this example is B. Alaykum salam. Number 21. Which of the following is an ancestral characteristic in mammals? Ancestral characteristic in mammals, it exists in the ancestors and it exists in us today. Definitely not feathers. We don't have feathers today. We have hair. We don't have a tail. Not all of us have a tail, but we all have a backbone. So not all mammals have hair. For example, dolphins and whales, they don't have hair. They are mammals. They don't have hair. Okay. Number 22. Which of the following is a derived characteristic in mammals? يعني it's a characteristic that only exists today but did not exist before. Probably, I would say it's here. The opposite. Okay? Then let's fill up these structures. Issa, you need to be memorize the first table. Describe the three things, the three types of structures, the homologous, analogous, and vestigial. You need to be able to describe them very well. The homologous are similar structures with different functions in different species. فيكم تكملوا وتقولوا they are inherited from a common ancestor. Example, the forelimbs of humans, cats, whales, and bats. The same example that we gave in the PowerPoint, same as the picture. Or for the analogous structure, they are structures that perform a similar function, but they have a different structure. They are not inherited from a common ancestor. Example, wings of insects and birds. Whereas for the vestigial structure, remember, don't say it doesn't have a function. Just say that it has a reduced forms of functional structure and other organisms. For example, tailbone, appendix in humans, wisdom tooth, أو floating ribs, or whatever. Okay? Now, number 24. In the table below, write the letter of the description that correctly matches each listed evidence of evolution. Let's see. Here, remains of ancient organisms that are found in layer of sedimentary rocks. This is for what? This is definitely speaking about the fossil records. So the A is here. The reduced form, directly, this is vestigial structure. Comparing early pre-birth stages of an organism's development, this is comparative embryology. Common ancestors seen in the complex, this is D is comparative molecular biology. Definitely is comparative molecular biology. Okay? Number 25. The below images are related to evidence of evolution. Fill in the table that follows to identify the type of evidence shown in each stage. Number 1, definitely this is fossils, the fossil records. Number 2, this is since you're seeing two hands, similar structure, different function, this is homologous structure. Number 3, comparative embryology. Number 4, vestigial. Number 5 is comparative molecular biology, or you can say molecular biology is okay. But this is all you need to keep in your heads. Now, just pay attention to the different words that we can accept. Okay? Just make sure you go over these. Number 26. Which of the following best defines the study of distribution of living things on Earth? So for comparative molecular biology and geographic distribution, how they are distributed on Earth, please, you have to memorize this word, biogeography. Bio, yani living things, geography, yani the study of Earth. So where are living things distributed on the face of Earth is known as biogeography. Number 27. Which of the following best demonstrates how geographic distribution provides evidence for evolution? Now, So I want to see how geography, يعني how is the face of the earth giving me evidence for evolution. A. The same species of mammals are found on different continents due to continental drift? No, this doesn't tell me. بينما if it tells you, if it talks about how the earth was a puzzle and different animals on different matching pieces of the puzzle on the face of the earth, they are there, that's different. Then B, unique species of, for example, marsupials such as kangaroos are found only in Australia, suggesting they evolved in isolation after the continent separated from others. Yes, that is a correct answer. C, identical species of birds are found in both the Arctic and the Antarctic, proving that evolution occurs independently of geography. No, that's wrong. But they are simply identical species because they traveled from one place to another, مش because they evolved in different locations. The fossils of ancient reptiles are found in the desert regions, showing they adapted to dry climate over time. لا, the presence of the reptiles أو حتى الfish in the desert proves that this area used to be a sea in the before. مش because they evolved. So the only correct answer so this is wrong. The only correct answer here is B. Number 28. Which of the following correctly describes camouflage in polar bears? For camouflage here, camouflage is the ability to smell? No. Camouflage is the ability to dig holes? No. Camouflage is fast swimming in cold water? Of course it's not. This is adaptation. All of these are adaptation. None of them is camouflage. Camouflage is having white fur that can blend with the color of the ice. Yes, this is one example of a camouflage. Then, 29. In the table below, write the letter of the example that correctly matches each listed type of adaptation. Let's see. Here, it's telling you MRSA bacteria. MRSA is a type of resistant bacteria. Okay? MRSA bacterial infection becomes very difficult to treat. This is an example of antibacterial resistance, definitely. The Arctic hare, يعني الأرنب, rabbit, the Arctic hare has white fur that helps it blend in with the snowy landscape it inhabits. This is definitely camouflage. And the last one, the harmless scarlet king snake has evolved its appearance to look like the venomous coral snake. Definitely this is mimic. Okay? يلا. Question 30. A large population of salamander is living in a certain ecosystem, and individuals of this population are mating randomly. List 3 other conditions for the population of salamanders to stay under the Hardy-Weinberg equilibrium. We have, in general, we have 5 conditions. Here it's telling you a large population, okay, or random mating. So he took 2 out of the 5. So all the 5, you should remember them. There should be no gene flow. This is what we have studied. But if you use other words, you're fine. If you say no immigration, no immigration, no migration, you're good. Okay? But no gene flow is fine as well. You can say no mutation, no natural selection. Okay? Now, but sorry, what I want you to pay attention for, we have 5 conditions for Hardy-Weinberg you have to memorize. Number 1, large population. Number 2, random mating. Number 3, no gene flow, no mutation, and no natural selection. Okay? Now, how to apply Hardy-Weinberg equation? Remember, we have the Hardy-Weinberg equation. This is 1. And we also have P plus Q equals 1. Okay? So P plus Q equals 1. First of all, number 31. In a population, the dominant allele frequency P is 0.02. So he is telling you that P equals 0.2. Okay? Which of the following is the frequency of the heterozygous dominant genotype? شو يعني heterozygous؟ يعني capital A, small a. Heterozygous. Okay? So what do we use to know the heterozygous here? We should use this one, 2PQ. So to know this, I need to calculate 2PQ. So do I know the P? Yes, 0.2. Do I know the Q? I can calculate it. Q is 0.8. How did I know? Because P plus Q is 1. So what I need to do here is to multiply 2 times 0.2 times 0.8. The answer is 0.32. So now if you come here, 2 times 0.2 times 0.8, this is times 0.2 times 0.8, the answer here is 0.32. So the correct answer here is 32, 0.32. So this is the frequency of the heterozygous genotype. Okay? Number 32. In a population of rodents, the frequency of the dominant allele is 0.4. So the frequency of the dominant allele is 0.4. What does this sentence mean? It means P equals 0.4. Which of the following represents the frequency of the homozygous recessive? يعني homozygous recessive, he wants to know the Q square. Do I know the P? Yes. So can I know from the P? Can I know the Q? Yes. Q equals 1 minus P, which is 0.6. So Q square is 0.6 square, which is 0.36. So I found the correct answer here. So the correct answer is 0.36. Okay? Now, let's continue. A small group of frogs became isolated from other original populations on a small island, which resulted in a decrease in genetic diversity. So he's telling you here, we have a big group of frogs. We took a small group out. So this is an example of genetic drift. Okay? Now, when a small sample of population settles in a new location separated from the rest of the population, this is known as founder effect. Okay. Now, I think the last one the last one is an example about bottleneck effect. A small population of cheetahs is experiencing a sharp decline in its size due to habitat loss. So there is a natural disaster here. Natural disaster, definitely this is an example of a bottleneck effect. So far so good? Number 36. Which of the following best explains how mutations affect allelic frequencies? Mutations only decrease allelic frequencies. غلط. Mutations can increase or decrease allelic frequencies. So decrease غلط. Increase is also wrong. Also, it has no effect. Definitely, it is wrong. Mutations always will have an effect on allelic frequencies. It can either increase or decrease allelic frequencies. Okay? Now, here, this is something that you have to memorize the 3 graphs, if you remember them. Directional or disruptive stabilizing. Here, the first one, so we have stabilizing, we have directional, we have disruptive. Last one is sexual. That's something that doesn't have a graph. The graph below illustrates a type of selection that is called, now as you can see, this is the original population. The dotted line here is the original population. Now I can see that the middle of the line is increasing, or the centers of the line, the corners of the line, the extremities are decreasing. In this case, this is something that I call a stabilizing selection. Okay, when the middle increases, will corners or sides decrease, this is called the stabilizing selection. Now, as you can see here, in the original population, when the middle decreases and the corner here increases, it looks like it is moving. This is something that I call a directional selection. Okay? Next, there is more about this. With the three graphs below show three types of selection. Complete the table that follows to identify the number of the graph that matches each listed type of selection. Each number can be used only once. So for the disruptive, definitely disruptive is the last one. Here is the disruptive. The directional selection when it's moving to one direction. Here it is. With stabilizing when the middle increases or the corners decrease. So 3, 2, 1, 3, 1, 2. Now, in this KPI, we will be talking about isolation. There is two types of isolation. There is a pre-zygotic, yani to prevent the zygotes from forming, to prevent the egg and the sperm from meeting, or post-zygotic, yani to allow the zygote to form, but to prevent the zygote from having babies. Okay? So let's read them quickly. The definition of the pre-zygotic, it occurs before fertilization. This one occurs after fertilization. Description, it stops mating and fertilization by creating barriers in location. For example, there is behavioral barriers, ecology, and reproduction timing. أو ببساطة you can simply say in the pre-zygotic, no zygote is formed. But it's enough to say this. In the post-zygotic, لا, there is a zygote that is going to form the hybrid, but the hybrid cannot survive or cannot reproduce. It is infertile. It is sterile. That's very important. So remember, I will write it here for you. You can simply say the offspring is sterile or infertile. It's a full answer. Now, how does it contribute to reproductive isolation? Pre-zygotic isolation, it keeps genetic material from entering a population's gene pool, and the post-zygotic isolation, it prevents the development and reproduction of hybrid offsprings. If you remember, for example, the mule is a post-zygotic, okay? بينما بالpre-zygotic, one very major example, if you remember, we did speak about mosquitoes. There are mosquitoes that only mate at the beginning of the night. There are mosquitoes that only mate at the end of the night. So they have completely different timings. They cannot mate together. Okay? Question number 41. Let's see. The diagram below shows two species of squirrels. I will make it bigger. The K-pap and a bird. I don't care about the names, but I know this is, you can see the pictures here. You can see their names that live around the Grand Canyon. They live in the same area. Over time, they diverged from an ancestral species. So he is telling you here, this is an example of two species that diverged from a common ancestor. Use the diagram to identify the type of speciation that is occurring in the mice population. Now, do we have allopatric or sympatric? You have to remember, because you can see them here, this is an example of an allopatric speciation, 100%. How did you know this is allopatric? Because they were separated by a geographic barrier. In the picture here, you can see the first squirrel is living on this side, the second squirrel is living on this side. So there is a barrier between them. So this is an example of an allopatric speciation. Okay? Number 42. Which of the following statements correctly distinguishes between gradualism and punctuated equilibrium? Okay, let's see. Gradualism suggests evolution occurs in rapid bursts. غلط. From the name, gradual يعني bits by bits, exactly. The second one, gradualism describes a slow, continuous process of evolutionary changes, while equilibrium suggests long periods of stability interrupted by short, rapid changes. That is very correct. So B is the correct answer. Now, let's move to a new lesson. But I will solve this, so I will stop for today. Okay, quickly. The table below shows the classification of a species of firefly. Complete the table by filling the empty cells in the classification taxon column. Now, name this taxon. Now, what is the big? I'm telling you that the firefly is in eukarya. What is bigger than kingdom? Domain. Yes, domain. I will just show you the words. Then after domain, you have kingdom. After kingdom, you have phylum. And then you have class. Then you have order. Then you have family, genus. And the last one is a species. Okay. And that's something that has to be given to us. How did I know the genus here? The genus is found in the table. Where is it? It's over here. Okay, so the first word is the genus. It has to be capitalized. It has to be italicized in italics. And the second word is the species, and it has to be smaller letter. Remember, if you are writing this in the exam, you have to underline it. Okay. Now define the word taxon. It's very easy. taxon شو يعني taxon؟ قلنا نحن تتذكروا بالبيراميد قلنا we have 8 taxa, 8 taxons, 8 different levels. ما بنقول taxons بنقول taxa. taxa بس named group of organisms. Okay. يعني a domain is a taxon, a kingdom is a taxon, a phylum is a taxon, class is a taxon, order is a taxon, and so on. Family, genus, species, each one of these is a taxon. Okay. طيب, let's stop here. Time is almost finished.